Alrighty, let's get started. Hi, everybody. Welcome to Connecticut River Conservancy's live stream. We're in season 2, episode 3, Water Monitoring Behind the Scenes. My name is Stacy Leonard, and I'm the events coordinator here at CRC, and we're really excited to have you joining us today. LiveStream is now our primary venue for bringing our work and our rivers to you wherever you may reside. You can find all of our past and future episodes on our website at ctriver.org/livestream. And I'll be sharing that link and all the links that you hear in today's presentation in a follow-up email afterwards. Just 2 quick details before we launch. We really encourage your questions and ask you to use the chat box, and we'll field questions at the end of the program. And we are recording this for later access. And that link gets posted on that same live stream website, and it will be in the email that you receive later today. All right, so for today's presentation, this behind the scenes look at CRC's water quality monitoring program will be guided today by Ryan O'Donnell, CRC's water quality monitoring coordinator, who's been leading this charge since 2013. We'll learn more about river pollution like bacteria, nutrients, and plastic, how CRC tests for it, and what we do with the data. Ryan will review the 2020 monitoring season, including Samplepalooza, our one-day 4-state nutrient monitoring event. And he'll give us a sneak peek into our new Microplastics monitoring program, which is very exciting. So I'm going to turn it over to Ryan now to lead us on the way. Thanks again for being here.
Hello, everyone. Thanks for joining us. This is my behind the scenes look at the water quality monitoring program that I run for Connecticut River Conservancy. I find that one of the questions I get a lot is, well, what do you actually do for your job? So this is my attempt to explain it and give a brief look at a whole bunch of things. So first, I'm going to go through kind of what's behind designing a monitoring program, what you monitor, where, when, and then what do you do after you've done the monitoring. A quick look at both Is It Clean / Está Limpio, our bacteria monitoring program and sharing network, and then also Samplepalooza. And then, of course, looking forward to this summer, our newest water quality monitoring program will be Microplastics, and then a little bit about how you can get involved. So first, starting with designing a monitoring program. What, where, when, then what? If we're starting from the beginning, what are we going to do? So first, you're probably going to decide what it is that you want to monitor. So this is going to be informed by what the main question of your project. What is the purpose? And what's the question you're trying to answer? And then once you know that, you can pick out what you're going to monitor for. So I have a selection of some of the things, although you can monitor for anything you can test for in a lab and then some. One of the most popular ones is E. coli bacteria, and that gives us information on recreational safety. So what is the risk of contracting a waterborne illness if you go swimming or boating in a particular area? And then it also tells you, is there any sort of fecal contamination? That's where you get most of the E. coli bacteria from, the waste of animals or humans. So maybe you want to test for nutrients so that you can see if there are any agricultural impacts. So, farmers or gardeners or golf courses, they all spread fertilizer maybe to improve their plants' health. But if the plants don't take it up, it often gets washed into a nearby river. And so too many nutrients in your river, your lake, your ocean can cause algae blooms and stuff like that. The nutrients are also found in septic and sewage effluent. So the water, even though it's been treated by a wastewater treatment plant or your septic system, one thing that they don't necessarily take out is the nitrogen and the phosphorus. So you can see what the impacts might be. Maybe you're concerned about road salt and its impacts on your waterways. Then you might be testing for chlorides, because that's part of the salt. If your water is maybe super cloudy, or there's some erosion that you're seeing, you would maybe test for turbidity, which is how cloudy water is. Total suspended solids is just the amount of solids that are suspended in water. Or Secchi depth, if you are from the lake world, that's a neat little disc that's black and white, and you kind of drop it down in the water and see how far down you can still see that disc clearly. And that's also a measure of how cloudy the water is. Maybe you want to know about how the habitat suitability is for your river, or if there's any thermal pollution, then you go straight for temperature. And then not something that CRC does a whole lot of, but different state agencies and some volunteer monitoring groups do biomonitoring, where they're actually looking at the fish or the macroinvertebrates, which are bugs, crawfish, little creatures that live in the sediment and in the water column. And that will tell you a lot about habitat suitability, and then also what the actual impacts of local pollution are. And like I said, there's plenty more things you could test for. You could test for metals, or PCBs are a big thing now to test for. So lots of different things, anything you can test for in the lab. But you'd pick what you're monitoring by what question you're trying to answer. All right, so then next, where do you want to monitor? So you want to look at, you want to select the locations, once again, to help you answer your question, to achieve your objectives that you laid out. So if you're testing for swimming, you want to test where people are swimming. If you're trying to identify a source of a pollution, so if you were testing your agricultural impacts, maybe, you would actually want to bracket your location. So you would select one site upstream and one site downstream. But then you also want to consider what else is different besides maybe the farm or the golf course you're trying to monitor for. Is there a stormwater outfall near there, a pipe that's carrying water from the streets? That might also affect your results. And are you far enough upstream or downstream to accurately document the issue? If you're too close, you might be getting kind of like almost like chunks of the pollutant. You want to be downstream enough that it's all well mixed, so you're seeing what the actual impact on the river that you're monitoring for is. And is the spatial resolution appropriate? Do you have enough sites for your project? Do you need more? Do you have too many? Are you spending too much money monitoring? And then one of the biggest challenges for where to monitor, especially for me working with volunteers, is there safe, consistent access for volunteers? Safety is our number one priority, and we want to make sure that we're not risking injury for anyone to collect samples. And sometimes it just becomes impossible. We scout out, I'm often scouting out new sites, probably in a month and a half from now, the snow has melted, the Japanese knotweed has not come up, or that poison ivy patch isn't fully full-fledged. And so sometimes I pick out a site, and then, June or July, that site becomes inaccessible and we have to figure something else out. So, and then here for my pictures, I have 2 years. In 2016 and 2017, I did a temperature monitoring project. And the first year, in 2016, as you can see, I I picked the sites basically looking at a map, and I thought these are well spaced out. And then went later, after I was processing the data, I was looking at the watershed and realized that this whole yellow area is between, you know, two sites, and it's like a huge amount of the watershed. And so I wasn't actually capturing a proportional amount of the watershed. So then the next year, I was able to add a few more sites and get a better, and break up the watershed into smaller chunks that were maybe more proportionate. We could see the effects of the different areas of the watershed. All right, and when are you going to monitor? You know, does the parameter change throughout the day? So here I have a graph of just generic dissolved oxygen. And if you're monitoring for dissolved oxygen, it's really important that you go out at the same time every day that you're monitoring, because you're going to see these fluctuations from sunrise to sunset based on, you know, photosynthesis of the aquatic plants. Maybe it changes after a rainstorm. So with bacteria, we see huge influxes sometimes after rains. And so if we're tracking down a source that we think might be related to stormwater, to the rain falling and getting into the river, we'd want to monitor after a rainstorm. Does it change with the flow level? So maybe you're trying to catch something that is abundant in the spring after the snow melts. So you want to be monitoring, you know, in March and early April, or same thing seasonally, you know. So you would pick when you want to monitor based on things that you need to know about the parameter. And then also there's some other things to take into consideration, like holding times. So for bacteria, for example, we often test in the morning, and there's kind of a tight window. We have 6 hours from when the sample's taken to when it needs to be at the lab, so that it's the most accurate. And it's really important to schedule the monitoring so that, you know, if someone's in the lab ready to process the sample, there's room in the incubator. So those are some of the logistics that go into thinking about when you want to monitor. And then you get back a lot of numbers, and they're very boring and hard to understand just to look at. And it's my job to then make it into something that looks more like the infographic on your right. So I took the 2020 bacteria results that were uploaded to our website that I'll talk about in a little bit, and I broke them down by wet weather or dry weather, and showed what the quality is. And so it turns something from that's just a block of text and numbers that means almost nothing to, and then we need to communicate that in a way that people understand what we're trying to say. So I want to be clear that, you know, after it rains, it is more likely that a site is going to be not clean for swimming or boating, or not clean for swimming, which I hope that this infographic portrays much more clearly than just sending out a block of numbers to people who are asking about the results. So that's my biggest secret. If you're trying to turn blocks of numbers into something interesting, is you want to use lots of color coding. So if things are bad, color them red. And if they're good, color them green, so that, you know, someone who's not used to looking at the numbers can easily look at a graph or a chart and see what's good and what's bad. All right. So next, I want to take a quick look back at 2020. Look at, is it clean / Está Limpio, and then Samplepalooza. So is it clean? Está Limpio is our bacteria monitoring network that is housed on our Is It Clean website. So you can find your nearest results, not just from us, but from all watershed organizations that monitor for E. coli bacteria, they are free to upload to this website, and then it'll color code it automatically. And then if you're going to the website, you can search by the city, the state, the water body, the monitoring organization, or the date. And it'll pull up your sites. And one thing that we're doing is that we're working to include statistical information to help bridge the gap between sampling days, because something about water sampling is, you're taking one bottle of water, often 100 milliliters or less, and it's just like a snapshot of the water for that day. And as you know, a river's always flowing, the water's always changing. And so even in the 24 hours between when we sample and when we get things uploaded to this website, things could change. And so we're starting to look at statistically how a site acts, either whether it's been dry for the past 1 to 2 days, or if it's rained significantly in the past 1 to 2 days. Because like I said, we see more bacteria after the rain. And our goal with this website is we want swimmers and boaters to be able to make informed decisions about the risks of recreating in our waters. So a little bit about E. coli bacteria. Like I mentioned, it's used to determine the level of risk for recreation. The levels for primary contact, which is something where you're in the water, you're likely to get the water in your mouth or on your mucous membranes, is set by the EPA at 235 organisms per 100 milliliters. And secondary contact is something like boating or jet skiing, where you're not in the water, but you might get sprayed, and then so it is less likely that you'll get a significant amount of water in your mouth, but it's still possible. And they set that level about 625 organisms per 100 milliliters. And this is what we call an indicator organism. What that means is the E. coli is pretty abundant, as you can see, 235 or 625 in 100 milliliters is a decent number, but it's abundant, and most of them won't make you sick. As we know, some E. coli can make you sick, but not all, but most doesn't. But since it indicates that the water has been contaminated with human or animal waste, there's a higher likelihood that there could be other waterborne illnesses that are harder to test for, and maybe you only need to encounter one or a few of them in order to get sick. And we see these increases from agricultural and urban runoff, failing septic, leaking sewer, and then something that can be really big is actually pet waste, people not picking up after their dogs while they're walking near the river, or even wildlife. And we see increased levels after rainfall. That's if you learn one thing from me talking, that's the one to learn. So a little bit about our monitoring program is that our volunteers collect weekly E. coli samples at popular recreation sites along the Connecticut River in Massachusetts and Connecticut, and then a couple tributaries. Our Massachusetts samples are tested at our lab in Greenfield, and our Connecticut samples are tested by USGS in Hartford for us. And there are also other organizations around the watershed collecting samples weekly, bi-weekly, or monthly, and those are also posted on the website. One concern in our area, especially with the main stem of the Connecticut River, is combined sewer overflows. So some cities have what are called combined sewers, where their stormwater, so like the storm drains and all of that, are connected to their sanitary sewers. So that's the pipes from your house, from your toilet, or from industry. And what that means is that all the rain that falls enters the storm sewer needs to go to the same place that all the water from flushing your toilet or doing your dishes is going, and it needs to be treated by a wastewater treatment plant. But when there's a large rainstorm, you have a huge amount of volume coming through, and some of it overflows, and the system is designed to overflow. You can see in this diagram on the bottom how it works. The system's designed to overflow, and raw or partially treated sewage mixed with rainwater, so it is diluted, needs to be discharged directly into the rivers. But luckily, yay, there's a recent bill just signed into law in Massachusetts that residents will now be alerted when these overflows happen. That is something that was not true before. So now we will understand even better how frequent these overflow events are. So looking at the results from 2020, all of our main STEM sites. First, we'll look at Massachusetts. We have the yellow dotted line is the swimming threshold, and the red dotted line is the boating threshold. The light green line is the average during dry weather for each site. And then the dark green line with the circles is the average during wet weather. Then down across the bottom, I put in where the 4 major tributaries come in Massachusetts. And then the red triangles are actually a combined sewer overflow location. And as you can see, in general, the numbers are pretty low in both wet and dry weather. But when we get into this CSO area, which is also where there's a lot of pavement and other impervious surfaces. So it's not just the impacts of these combined sewer overflows, but it's more developed as we're getting into Chicopee and Springfield. You can see that these numbers are really high, particularly in wet weather. Yeah. And then if we look at Connecticut, we start monitoring in East Hartford and going down from there. And you can see these numbers are actually really, really low, which is good. And it's in part because there's just, by the time we get in Connecticut, there's so much water in the Connecticut that all of that bacteria from further upstream is very diluted. All right, so now a little bit about Samplepalooza, which is our one-day synoptic nutrient monitoring event. What that means is that everyone goes out and they collect samples all on the same day, all using the same method. They're collecting for the same parameters. They're all being tested by the same lab, and it's quite an undertaking. So in 2020, we collected samples at 69 different locations in 4 states. We collect samples both from the tributaries and then also the Connecticut River itself. So our goals are to assemble a really unique snapshot. So remember, like I said, these are snapshots. You know, this is like if you were taking one picture once a year, and then this is a huge geographic area for a study like this. And another goal is just the collaboration that's involved in getting this done. And then also to collect quality assured data. That means that, you know, if the states or the EPA or anyone wants to use our data from Samplepalooza, they are confident that the numbers are correct, they're precise, they're accurate. And so that is very important to us. One of the challenges is supply distribution and sample transportation. We had to get bottles to all those people before the day. And we also had to make sure that all of the samples ended up at the lab by the end of the business day. So that's always a long but rewarding day of my year. So, and then the other challenge, as is always a challenge, is interpreting this data in a meaningful way, especially because we cross so many political boundaries. And I just want to give a shout out to all the partners that make Samplepalooza happen. I also like to call this page the collage of blue and green logos. But all of these organizations and agencies contribute volunteers or input on where to collect and or help with transportation, everything. You know, certainly couldn't do it all by ourselves. All right, so when I'm looking at the results, this was one of the most challenging things because each state has different standards, and most of the states actually don't have a standard that has a number in it. So we're getting back a number, and how do we definitively say, is this number too high or is it an acceptable level? And even though some states do have numbers, it turns out other states don't like it when you use other states' numbers to judge their data. So when looking for a federal guideline, and luckily the US EPA, the Environmental Protection Agency, has developed recommendations for nutrient levels based on the ecoregion, and this is a picture of part of the map of the ecoregions. And so the Connecticut River watershed is in 2 of them. So that's in the nutrient poor and largely glaciated upper Midwest and Northeast, and the eastern coastal plain. Anyway, so this is just a first look, and these are all the tributary sites, north on the left and south on the right. And if they were, the watersheds are green, the nitrogen levels met that ecoregion recommendation. And if they're red, they exceeded that recommendation. And then if the background is darker green, that means that it's in the upper ecoregion, and if it's lighter green, it's in the lower ecoregion. So you can just get a quick peek at the total nitrogen. And then here's a quick peek at the total phosphorus. In general, there are more red watersheds for phosphorus than for nitrogen. But we really care about these because, you know, all of our water is ending up in Long Island Sound, and they have too much nutrients, particularly nitrogen, contributing to algae blooms and other problems with eutrophication and fish kills and stuff. So here's a look at our main stem. In 2020, the main stem did really well on total nitrogen, which is great, but really struggled with phosphorus. So one thing is, you might have noticed that the tributaries are all different sizes. So when we get our results back, they're actually in concentrations, which is the amount per an equal amount of sample, but the tributaries are all carrying different amounts of water. And so we actually want to look at also the load, which is basically how much is contributed over time. And so we need to kind of also take into account flow. And so this is the part where I'm a little ahead of myself. So I haven't done these measurements for 2020, but I wanted to give you a quick look at, this is 2014, 2015, and then 2018 and 2019. So these tributaries are ordered by size of watershed, and then the bars are their loading. And one thing that we can see here is what tributaries are having a disproportionate impact on their of the loading. So for instance, if we look at the Scantic, we would expect this, as they get smaller, the loading, they get smaller and smaller. And the Scantic really stands out here as contributing a lot of nitrogen. And so we would want to go back and maybe, do more investigation there. So the 2020 flow calculations are in process. We have to run it through a program that was put together by one of our volunteers. And so that will be coming. So this is just a very first look at Samplepalooza, and keep your eye out for a full report coming in the next couple months. So 2021, really excited about this microplastics program. So microplastics, you may have heard about them. So there are, different types of microplastics. they're all small particles of plastic that are, degraded larger items like styrofoam, bags, bottles, wrappers, synthetic textiles. They're really small, less than 5 millimeters in diameter. And then these are some of the categories of microplastics. So we have fibers, pellets, foam, film, and then just various fragments. And these are all found, in our rivers and oceans, unfortunately. So there's been growing concern in recent years about the amount of plastics in aquatic environments. Here's a picture from Scientific American of a beach just covered in little pieces of plastic. And the plastic, trash can range in size from, whole boats full of trash to microbeads and glitter. And the real problem with plastics is that they last, almost indefinitely, and they're very hard to clean up, and that we're finding that they're accumulating in the food chain. And as we know, all of our water is ending up in Long Island Sound in the Atlantic Ocean. So any plastic that's in the Connecticut River is ending up, in the ocean. And so that's why we really wanted to start looking at what our watershed is contributing. So just a little peek into the process of what we do in the lab. So we collect a sample. I actually have one right here waiting to be processed. Collect a sample, and then you kind of pump it through this vacuum filter thing, pull the water through. You get a sheet of paper that will have everything that was in the water on top of it. Look at it under a microscope, and then we're actually going to count the plastics that we find. So right now, we collected a few pilot samples, me and some of the other staff. We're working on creating standard operating procedures and a volunteer manual. And then we're going to start training volunteers so that you all can get out there. And so the microplastics grant is funded by the Massachusetts Environmental Trust. So if you have one of these fish or whale license plates, you're helping fund this project. Also the Conservation Law Foundation and the International Coastal Cleanup. So this is the first microplastics monitoring program in the region. They're in New England. There's some in the Hudson and in the Great Lakes. But one thing we're really excited about is this really is connecting our community science water quality monitoring with our source to sea and trash advocacy. It's really building a bridge between different program areas. And also it's our goal is to create an adaptive protocol that collects quality data to this emerging field of science about our watershed, add to the variety of community science volunteer opportunities that we offer, and fill in a major gap of knowledge about what's in our rivers with regards to plastic. Because right now we don't know. So, there's a lot going on, and I want you to get involved. So some 2021 water quality monitoring opportunities. You could become a bacteria monitor in Massachusetts or Connecticut. You could volunteer with your local watershed organization. They're scattered all over our 4 watershed states. Sign up to collect microplastic sampling, or perhaps volunteer in the lab to process the samples. Samplepalooza is coming in September. Something that we often need help, it's a little less glamorous, is actually with the sample transportation. Driving those samples around is something we often need help with, and many more. And so you can visit our website, and Stacy will send that out to you. And without further ado, thank you so much, and I'm happy to answer questions.
Great. Thank you, Ryan. That was really thorough and full of lots of details. I'm sure people have questions. So want to remind folks to please put your questions in the chat, and we may, if we get through that, be able to just raise hands. But I thought it might help just to clarify a little bit why or what's happening in Vermont, New Hampshire. Are we not CRC not sampling there, but people would go to a different watershed association if they want to participate there?
Yeah, so currently we don't have a bacteria monitoring program on the main stem in Vermont, New Hampshire. there are some groups that do, for instance, the Putney Rowing Club monitors at their docks. Trout Unlimited has at times done a site in kind of the Rockingham area, but currently we just don't have that program going on. But there are plenty of watershed organizations also doing tributary monitoring. We're really limited, like I said, by the holding time. You know, we have 6 hours from when the sample is collected to when it is processed, so that we can, that's why our Connecticut samples go to Hartford and not up to our lab, Greenfield.
Okay, great. And also, with that big impact of the CSOs that you showed in the diagram, can you talk a little bit more about, you know, what's being done about that today to address the CSO overflows?
Yeah, it's not something that I am super expert in. I don't know if I see any of my colleagues on here who know a little bit more about it. But for instance, I know that Hartford has been working really hard to address the CSO. All the cities that have it should be somewhere in the process of either separating the storm sewer and the sanitary sewer, or working to create something where they like store the excess water somewhere so that it can then be treated later. So I don't know the exact process that that's going on right now in Springfield, or where they are in the process. That's definitely something I encourage people to learn more about. But it's something that is being addressed, but is somewhere along the process.
Great, thanks. So a couple of questions. Somebody was asking about where can volunteer groups acquire funding? You talked about the microplastics funding, but maybe more broadly on the.
Yeah, funding water quality monitoring is difficult. It's not always the top priorities of funding organizations. Something that we, you know, in Massachusetts, the Department of Environmental Protection has been supporting certain types of monitoring in the past few years. Certainly Vermont and New Hampshire each have kind of their state programs that are very limited in like what parameters you can collect and where you can collect them. But yeah, there's I'm still trying to figure that out sometimes, how to best fund it. One thing that we do work to do sometimes is we've had, you know, businesses sponsor like a swimming hole if we're doing that bacteria monitoring, and we can post their logo and email on their website or on any like posters we might put up at the site. But yeah, that's definitely a big challenge of this, this job.
Always. There was a little confusion about which Pioneer Valley, was it rowing club or yacht club? That is one of the testing sites, just if you could clarify that. Or
Yes.
maybe both.
Both are sites.
Okay.
So the Pioneer Valley Rowing Riverfront Club is the upstream one, and then the Yacht Club is I believe it's our furthest downstream site in Massachusetts.
Great, thanks. So here's a question from Noah. Do you foresee climate change and more severe weather patterns bringing more pollutants or new pollutants into rivers? Any other anticipations or preparations regarding climate change?
Absolutely. As our weather gets more erratic and we're seeing larger rainstorms, or particularly more rain on snow events in the winter, or tropical storms making it up here, that's a big issue for water quality. For example, when Tropical Storm Irene came through, and it activated a lot of landslides. It caused a lot of landslides. And so the thing about landslides is there's very little you can do to stop them. You're just going to have to wait for them to run their course. But our soils have a lot of the nutrients in them. That's why plants like to grow in them. And so they're very laden with phosphorus. And so what we see is even these very, undeveloped areas like the upper Green River watershed. Now, every time it rains, every time we get a big rainstorm in the summer, there's a ton of sediment coming from those landslides and carrying with it phosphorus.
Oh, good, you unfroze.
I know, I saw everyone freeze, but I hope you heard what I said.
Yeah, great. David asks, are there any town or city regulations controlling upstream pesticide, agriculture, or fertilizer runoff? Is this a state or federal role?
I think that there are a lot of state regulations, particularly in New England, for what the impacts are allowed to be of your agriculture or fertilizer runoff. I know that in Vermont, for example, they have kind of required management practices. And if your farm is contributing too much runoff that they can document, they're going to require you to to implement some of these, or they might come by, it might be part of your inspection to make sure that if you're a dairy farm, for instance, that you're storing your manure, not in a pile next to the river. So there's definitely regulations, and it's different in each state.
Great. Rob says, Springfield has recently done extensive storm drain work, multi-year projects, which had bike path locations closed along the river for a long time. Has there been any noticeable difference in water quality since completion? That's a neat question.
That's a great question. It's not something that I've looked into. We just took over those Springfield sites. The Pioneer Valley Planting Commission used to run the southern portion of the valley, the monitoring program there. But I definitely have access to that data. That's something I could look into. It's very hard, to kind of incidentally catch something like that. Like I said, we would probably want to be bracketing and sampling specifically after rainstorms, but it's possible that we could go back and look at weather data and stuff like that and see if there was any difference.
Great. So maybe someone could get in touch with you if they want some.
Yeah, that sounds like a fun
detail.
project. I could set you up for a volunteer project. I could definitely set that up.
Great. Hey, I see a hand from Sky, so feel free to unmute. And also anyone is welcome to put your video back on and raise your hand, and we'll do it this way. So please, Sky.
Hello, my name is Sky Lloyd, and I'm from Hardwick, Massachusetts, on the west branch of the Ware River. And my question was if there are any. Actually, I have two questions. If there are any testing sites on the west branch of the Ware River, and my second question was, are the smaller streams, the tributaries to the Ware River, tested as well?
The Ware River is a tributary to the Chicopee, right? So the Chicopee Four Rivers Watershed Council has a pretty extensive monitoring program. So I'm trying to pull up the Is It Clean website to see. But you could check the Is It Clean site to see if there's any monitoring going on there. But then also USGS right now is doing a project with the Pioneer Valley Planning Commission in the Chicopee watershed. I don't remember if the Ware River is one of their sites, but they're really they're looking at kind of what the different land uses are contributing pollutant wise. So that's not a volunteer opportunity, but it's something to kind of keep an ear out for once that project wraps up and what they put out there. But definitely check with the Chicopee Four Rivers Watershed Council.
Thank you.
Does anyone else want to pose a question? What brought you here? What's on your mind? Jamie, please unmute.
Yeah, thanks for doing this, Connecticut River Group. So I'm thinking as we're going to be doing more measuring of microplastics, what is the remediation possibilities?
Yeah, that's a great question. I know that some of, once the plastic's in the river, I think it's too late, right? But I've seen some of the remediation approaches, kind of prevention approaches, where you can put these kind of like nets on your stormwater outfalls that could catch larger plastics. I saw Gabriel turn her camera on. I don't know if she has something to say. No, she's helping me run the microplastics program. But so yeah, it's all about prevention and making sure that we're not letting our plastics end up there.
There's also some great information from some other groups that have been doing microplastics works. I think there's some in Canada or in the Midwest. I have to go put my fingers on it, but they're trying to advocate for people to put filters on their washer dryers to prevent all the plastics that are in our clothing from entering the water system. So there's so many levels of places to get involved in this. And of course, our Source to Sea cleanup is a place where we literally go in and pick up all the trash that we find. And there's just, as you can imagine, tons of plastic waste every year. I'm going to dovetail over back to the chat. Lynn is asking, I have a great site on the Connecticut River in East Deerfield with 1,400 feet of frontage and great access. She wants to volunteer it as a test site. So what is involved in adding a test site to the docket?
Yeah, usually we're looking for a specific place. We test, I guess we don't test there. I was thinking we test the Deerfield in East Deerfield, but not the Connecticut River. Yeah, so definitely send me an email. A lot of it's dependent on funding and also though thinking about like the questions that we're trying to answer. So a lot of our Connecticut River sites are at docks, at rowing clubs, Bart's Cove, places where it's a public access where a lot of people are going to be, but that doesn't necessarily rule out, if we're missing a spot along the river, which we might be. So feel free to send me an email and we can talk about it.
Great. There's another question in the chat from William. Is there a way to compute the cost of inaction to help get more steam behind this effort? Woo.
Yeah, that's a great question. I'm certainly not an economist by any means, but that is kind of one of the big problems, right, with environmental pollution is that we kind of don't include it in the cost. We don't include the cost of emitting carbon dioxide and the detriment to our climate in the cost of a car or when you're buying, the gas for your car. But maybe we should, because right now we are saying it's free to pollute your water or your air. And so definitely calculating the costs is very important. I know for like Long Island Sound, where they're having, there are people who are calculating the amount of revenue lost because fisheries aren't able to operate. Or for example, in I feel like I went to a webinar recently where they were discussing the tourism money that you lose if your swimming area in your town is closed due to an algae bloom. So there's definitely ways to calculate the cost of what's lost by having water polluted, and the cost of course, prevention versus remediation, because preventing it in the first place is always cheaper than going back and trying to fix the problem later.
Kate asks, how do we sign up to volunteer? So that will be sent to you. It's Ryan's email, actually.
Yeah.
And we'll send it out again in the email that I'll send later today. And it was on the screen, and it'll be, so you'll see it in the recording as well. But rodonnell@ctriver.org. But again, I can put it in the chat, actually. Would anyone else like to ask a question? We probably have time for one or two more. I see Sky coming back. Don't forget to unmute.
Real quick, if we spot a like a CSO that has a broken, cracked pipe or something, we see, or like we're experiencing, like we see raw sewage pouring out, who do we call if we find, like there's been times I always explore small streams, and I've noticed sometimes there's CSOs that are like discharging raw sewage. So like, who do we call when we see?
You could you could contact us, or I've also had, I would do two things. I would get in touch with us so that because not me, but our river stewards know exactly who to get in touch with to report those problems, and we have relationships with those people. But then also you can report. In Massachusetts, there's definitely a website where you can go to the environmental police, I think, and report a violation. And I actually did this recently in the fall. I saw a place where a contractor had dumped a bunch of waste right on a stream side, like a bunch of like drywall and stuff. And I reported it, and they actually got back to me either later that day or the next morning. And a few days later, it was gone. So I found them to be very responsive.
Thank you.
Would anyone else like to ask one more question?
Can I just make a comment to Ryan's answer to Sky? I think that the double teaming of it and reaching it from those different points, if you have the capacity to report it online and to reach out to CRC or the river stewards, is helpful, because the volume of people complaining about something like that speaks to action sometimes. So I would say, report it, but then also, if you have the capacity, reach out to CRC. And then, if Ryan called and Andrea called. So I think that, it would kind of move it up the ladder.
Nice.
Absolutely.
Great. So I think that's probably about, well, is there one more question before we close? Well, great. It means that Ryan was utterly thorough, and everybody got their questions answered. So we really appreciate you coming out today, and likely you'll have more questions when you hang up the phone. So feel free to reach out with other questions as they come to you. And as I said, I'll be sharing an email with all the links, how to volunteer, how to get onto the Is It Clean website, other volunteer opportunities at CRC. And if you're interested in a copy of the slide presentation, you can email me, and my email will be in the email, and we can make that available to you. They're usually kind of large files, so it's hard to share otherwise. But we look forward to seeing you again at future live stream presentations. They're about once a month on selected Wednesdays. So hope you'll join us. Our next one coming up is March 31st. It's called Tales from the Trail, Exploring the Connecticut River Paddlers Trail. And Gabriel, who you heard from, who is part of our lab team, is also coordinating this big effort of the Connecticut River Paddlers Trail. So we'll be hearing from 2 people who have made full journeys on the whole Connecticut River, sharing their stories and learning more about this thing called the Paddlers Trail and how you can also be a paddler on it. And there's a new app coming out, so you can get all the resources that you need from the app. And that will be March 31st at noon as well. So thanks again. Thanks to Ryan for all the great research and presentation, and for all of you for being here today.
Thank
Hope to
you,
see
everyone.
again soon.
Thank you.